Welcome, Panagiota, and please.
Good morning. It's nice to hear all these presentations. So good morning from me as well. My name is Panagiota Mouraka, and I'm a PhD student in the University of Nottingham. Today, I'm going to present you my research in the comparison of the physicochemical properties of different plant cell wall materials. And here is the outline of my today's presentation. So in the beginning, I'm going to talk about the background, then the methodology, and in the end about my results. So let's start from the background. So the motivation of this research are the structural and nutritional benefits that occur when the dietary fibers are added in the food systems. For this reason, the food industry is trying to find new strategies for designing healthier products using plant cell wall materials for structuring food matrices. The dietary fibers come from various sources, for example, cereal, legumes, fruits, and vegetables, and they can incorporate it in different products to make them more nutritious, healthier, as well as consumer-oriented. There are several food categories in which dietary fibers have become an invaluable ingredient. Some of them are bakery products, cereals, pasta, dairy products, meat products, and beverages as well. But what are dietary fibers? To start with, dietary fibers are plant-based food structures. They provide a multitude of functional properties when they are incorporated in the food system. Some of the health benefits are summarized here. And for example, the intake of dietary fibers has been suggested to improve glycemic control, lower lipid levels, they exhibit prebiotic effects, and may be a useful tool in weight management. Furthermore, addition of dietary fibers to food products contributes of the texture, sensory characteristics, and shelf-life of the products due to their water-binding capacity, gel-forming ability, fat-limiting, and thickening effects. Dietary fibers are a complex mixture of polysaccharides with many different functions and effects as they are passing through the gastrointestinal tract. Many of these functions and effects depend on their physicochemical properties. Solubility, hydration properties, and oil holding capacity, viscosity, and microstructure are some of the physicochemical properties that are linked with the dietary fibers. It is important to mention here that the origin of the dietary fibers affects the physicochemical properties, and so the texture and then sensory properties of the food to which they are being added. Therefore, since different fibers have different functionalities, a careful selection of the source or the sources most appropriate for the particular food is necessary. For this research from the cell wall structure insights, we will believe to target fibre engineering in all continuous food matrices. Just to summarize everything, the aim of the current research is the investigation of target engineering of fibre-loaded all continuous food matrices using plant cell wall derived from dietary fibers. The objectives here are the characterization of the physical properties of the dietary fibers and observation of their effect on the flow properties of the dietary fiber suspensions in sunflower oil. And in the hypothesis, we expect that the wettability of the dietary fiber particles affects the rheological properties of the concentrating suspension, in addition to size, shape, and mechanical properties. Moving now to the next section, I will be discussing the methodology that was followed in my research. The properties of wheat, oat, citrus, up, and pea fibres were considered in order to develop my methodology. More specific, in the following slides, I will be discussing the methods used to determine the respective fibres, microstructure, the effective oil holding capacity, which is represented here as the packing volume determination, and the flow properties after they have been dispersed in sunflower oil. But let's start to investigate each one of them in detail. Starting with the determination of microstructure, each sample was dispersed in sunflower oil, and the bright field and polarized microscope was used to observe the morphology of the suspensions. Furthermore, as discussed previously, the effective oil holding capacity is an important factor, especially in the continuous oil systems. It corresponds to the amount of oil that a sample can absorb. And in our case, it was measured by adding a specific amount of sample in an excess of oil, and then continue to the centrifugation of 2500 RPM at 200 degrees Celsius for 30 minutes. Afterwards, the effective oil holding capacity was determined as a function of the weight of the oil that was absorbed and trapped to the weight of the fibre. Finally, one very important factor in the properties of the dietary fibres is the viscosity, the flow behavior. And for this reason, the flow behavior of the various dietary fibres dispersed in sunflower oil were measured using two measuring systems: a concentric cylinder, as you can see here, at a decreasing shear rate from 1000 to 0.001 reciprocal second, at a gap of 1 millimeter, applying a pre-shear rate at 5 reciprocal second. And also, apart from the concentric cylinder, a parallel plate system at a decreasing shear rate from 100 at this time to 0.001 reciprocal second at the same gap of 1 millimeter, applying the same pre-shear rate at 5 reciprocal second was also used. All the flow behavior experiments were measured at 25 degrees Celsius at 30% mass concentration. Following my methodology, I will now present you some preliminary results. According to micrographs, as we can observe here, the microstructure of oat and wheat fibres is similar due to their origin from cereals, and they exhibit a fibrous morphology. Citrus and apple fibres from the other side are derived from fruits, and their morphology is irregular, as we can observe from the micrographs here. And finally, the pea fibre, which is here, which falls under the legume category, is full of starch. And we can observe that from the polarized micrograph at the bottom right corner. As discussed earlier, these images were taken from a bright and polarized microscope as well. Moving now to the effective oil holding capacity results. The wheat and oat fibres, they exhibit the lowest oil holding capacity, which can be attributed to the fibrous microstructure and potentially to their hydrophilicity, which we can see here. The fibres that are extracted from fruits, the citrus and apple fibre as well. So higher oil holding capacity compared to those that come from cereals. Similar behavior demonstrates also the pea fibre. These results confirm that the morphology of the various dietary fibres, as we observed earlier, affects the physical properties, and in this case, the effective oil holding capacity, which is an important factor that is considered in the food industry, since it is related with the texture and also the mouth-filling experience as well. The flow behavior of the different dietary fibres dispersed in sunflower oil can be seen in this graph. The viscosity curves of the different dietary fibers were examined using the concentrating cylinder, geometry and 25 degrees. A shear rate control test was carried out using ascending logarithmic steps. And we can observe here the wheat fiber, which is represented in the red color, and oat fiber, which is represented in orange color, exhibit shear-thinning behavior at 30% of their concentration. This can be potentially attributed to the fibrous-like microstructure and the fact that they are probably denser compared to the rest of the examined materials. Also, we can observe the citrus and apple with the light and the dark green color, and the pea fiber with the purple color, that they show substantially less shear-thinning behavior than with the oat fibers. This can be explained probably by the volume fracture and the irregular shape of the particles, as we observe from the morphology as well. And on the right-hand side, you can see the same results are expressed as the shear stress versus shear rate graph. And in order to verify my flow behavior results, I also ran some experiments using the parallel plate system to be more sure about the flow behavior of my materials. And as you can see from the graphs, similar flow behavior patterns are observed with the same shear thinning behavior with the wheat and oat fiber, which is quite marked. And the same as well, shear thinning behavior, but this is less shear thinning behavior compared to the wheat and oat fibers as well. And here again, you can see the graph, shear stress versus shear rate. Oops. Yeah. And here are presenting the key findings of my preliminary data, which is the fact that different groups of fibers are dominated by different materials, as we observed earlier. The fibers that are extracted from fruits show higher effective oil holding capacity compared to those from cereals, which may be associated with the fibrous microstructure and higher hydrophilicity of the latter. Due to highly elongated shape, wheat and oat fibers and specimens show a marked shear-thinning behavior, which can be attributed to the alignment of fiber particles in the flow. And also the apple fiber exhibit less shear-thinning behavior similar to citrus and pea fibers. And this is potentially due to soft nature of the plant cell walls, and also the absence of large cellulosic fragments, especially in the apple fiber. However, I have to take into consideration also the fact that the wheat and oat fiber, they were measured at 30% of their mass concentration, which is on the one hand is positive because then they can be used at lower loading. However, it's negative as well because they appear to be denser than the rest of the fibers, meaning that they're likely to form a residue on the oral surfaces due to more rapid sedimentation. And this is a general conclusion from my results as well, which indicate that the application of these dietary fibers in food formulation requires optimization of the particle size, their shape, and hydrophobicity to achieve a targeted flow behavior. And in the future, I will be investigating the flow profile at different concentration of my materials, the particle size distribution analysis in order to have the same size using size fractionation, either with sieving, and also the rapid visualizer in order to observe the changes as a function of temperature and the viscosity. Thank you very much for your attention, and I'm happy now to take any questions.